All right, hey everyone. I am going to do the same thing I did last time with the test review. I'm going to pull up the actual document and go through some of the questions. It's come to my attention that some of these questions actually are a little bit too detailed. So if you're watching this, you made a really good choice because I'm going to eliminate some of these questions. The first one is, what are the components of a nucleotide? That's not this slide right here. The components of a nucleotide, so you see, look at my cursor, you see this dotted blue line? This whole thing is a nucleotide, this whole thing. So the three components are a phosphate, a sugar, and a nitrogenous base. All right. Next question, and then A pairs with T, C pairs with G, and they pair together with hydrogen bonds. We've got DNA running anti-parallel. That's the third question. How is DNA anti-parallel? Well, the 5' end of the nucleotide is up on this strand. The 5' end is down on this strand. So if you kind of look at it, like the sugar is upside down right here. These are two strands of DNA, one on the left, one on the right, and they're bonded together with hydrogen bonds. So anti-parallel means they're running in opposite directions. What does that mean? Well, the phosphates are up on this side, the phosphates are down on that side. All right, so that's the third question on the review. What else? Okay. Second question, how is prokaryote and DNA and eukaryote DNA different? Sorry. There was that card sort thing that we did in class. I think H-Block didn't do it yet, but you're going to do it. H-Block didn't do it yet. But what I did was I gave them a piece of paper that had prokaryote DNA on one side, eukaryote DNA on the other side. And then all of these words were cut into little pieces. And so they had to sort them under which column do they go under. So I don't think there's really anything crazy about this. I just don't really have much to say except, both have double-stranded DNA. DNA is not DNA unless it's double-stranded, unless it is in a virus. There is single-stranded DNA viruses. I wish I knew an example off the top of my head, but I don't. But I will tell you when we get to the virus unit. I'll have a list for you. Prokaryote DNA is circular, eukaryote DNA is linear. Prokaryotes have one chromosome. Eukaryote DNA is usually more than one. Eukaryote DNA has histones. It's found in the nucleus. Y'all can read. All right. Semi-conservative replication. Remember you had a question on your quiz? Studying your quiz would actually be a really good idea. Like a really good idea. Semi-conservative replication is when you have the parent strand, it gets pulled apart, and then DNA is replicated on each side. So the light blue is the newly replicated DNA. So now you have two DNA strands as a result. Each one retains one of the parent strands. So each one of these new DNA strands has a dark blue and a light blue, a dark blue and a light blue. That's semi-conservative replication. I think that's there just to ensure accuracy, instead of the parent strand opening up and then making a brand new daughter strand, retaining one of each. So it serves as a really good template. I think it's to avoid mistakes, right? 7 steps of DNA replication. Boom, boom, boom. There they are. That's them. Read them. I would be able to put them in order. You could print out this list and put them in order. We had a card sort where we put the steps of replication in order. That was a good exercise. I think what's most important though is to know what each enzyme is. So I would definitely look at the enzyme list and what other jobs are. And they're listed for you. I think here, yeah, they're listed right here. I would make flashcards, know this list. Other thing that I wanted to go over was leading strand versus lagging strand. So so I'm on the review. 7 steps of DNA replication, name all the enzymes involved in replication and their job. You just get that from your notes, it's on there. Why can DNA polymerase only synthesize DNA in one direction? Because it can only add new nucleotides to the 3' end. It can only add new nucleotides to the hydroxyl group. Look at that. It's adding a new nucleotide. It's covalently bonding a new nucleotide to another one at that hydroxyl group, the 3' end. That's why it can only synthesize in one direction. So that is why Okazaki fragments exist, because DNA in the leading strand, so there is a question asking you, what is the difference between the leading strand and the lagging strand? Well, the leading strand is being replicated towards the replication fork. The lagging strand is being replicated away from the replication fork. Why? Because DNA polymerase can only add nucleotides to the 3' end of another nucleotide. It can bond a nucleotide only to the 3' end of another nucleotide. So it can move continuously towards the replication fork in the leading strand, but in the lagging strand it's moving away, and it's doing it in short spurts as DNA is being pulled apart and separated. So those are Okazaki fragments, or these short spurts. They can't stay like that though, so DNA polymerase 1 comes in, replaces primer, ligase seals them together, and now you have your beautiful new strand. All right, what is involved in proofreading and repair of DNA? I think it's just important to know that DNA polymerase proofreads and corrects as it goes. Then other enzymes come in and also proofread and correct. And I think that's really all you need to know. A nuclease is an enzyme that cuts out damaged DNA, and then DNA polymerase and ligase are going to fill in the gap. So it's just making corrections. That answers that question. Explain the significance of telomeres and telomerase. Ooh, yeah, I want to go over this. Okay. Eukaryote chromosomes are linear. That means at some point there's going to be an RNA primer at the very tip of the chromosome that cannot be replaced because DNA polymerase cannot add it there because of its inability to add to a 5' end. That primer ends up staying there and getting chopped off. So we have these things called telomeres that cap chromosomes. So if a little piece of a chromosome at the tip needs to be chopped off because there's a primer there that can't be replaced, that's fine, because we have telomeres that are just junk. I don't want to call it junk DNA. It's not junk DNA. It's just non-coding DNA. TTA, GGG, TTA, GGG, over and over and over and over and over again, capping. And so this picture is showing you chromosomes under a fluorescent microscope where you see the little orange things. Those are the telomeres that are the little caps of each chromosome. So they can get chopped off as replication occurs over and over again, and the primer at the very tip can't be replaced and gets chopped off. The telomere is the non-coding DNA at the ends. It can get chopped off and everything's fine. What I need you to know is what happens once all the telomeres, once it reaches the end of the telomere, it is chopped, 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 chopped. That cell is divided, or that DNA has been replicated so many times that the telomeres finally run out. Well, that is what's going to signal the cell to undergo apoptosis, which is cell death. So what organelle gets triggered? Lysosomes. Lysosomes get triggered. Remember that. I think it's kind of cool because I didn't ask a question about telomerase. There is a telomere question, but I didn't ask about telomerase specifically. But our next unit, we will be talking about cancer, and I will ask it again. What enzyme is it that adds telomeres back in cancer cells? So if a person has breast cancer, the cells of the breast tissue should not have telomerase in it. Telomerase should not be adding telomeres back in the breast tissue. It shouldn't be adding telomeres back anywhere except germ cells. And remember I said germ cells were the cells in the testes and the ovaries that undergo meiosis to form sperm and egg. If it's not a cell that's forming sperm and egg, it shouldn't have telomerase. But what has been found is that in cancer cells, telomerase is very active and is making sure that those cells never undergo apoptosis. That's not good. That's no good. Let me look. I think that's it. Yeah. Chapter 13, that's it. Ooh, 10 minutes. Y'all, this video is going to be shorter than the last one. Because it just is. Because it really is less material. And I think that oh shoot, where's chapter 14? Nope, that's not it. I'm gonna have to pause. I just think that that group quiz like really prepared you for this. Okay, I'm gonna pause and then I'll come back. Hold on. Okay, I'm back. So this is chapter 14. This is the next. Yep, chapter 14. I know on the top of your notes it says chapter 17. That is a typo. It was chapter 17 in my last book, but it's chapter 14 in this book. So, what is the purpose of transcription? Well, let me tell you. Transcription is transcribing DNA into RNA. It's converting DNA into a form that can actually leave the nucleus. DNA can't leave the nucleus. Well, in prokaryotes, DNA is just sitting there in the middle anyways. DNA just sits there. It doesn't like do anything. It just codes for stuff and it just sits there. It's kind of lazy. RNA is our real superstar here. RNA is going to move to a ribosome and it is going to be translated into a protein. So DNA to RNA is transcription. RNA to protein is translation. Boop, 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 boop. Okay, DNA and RNA are different from each other because they're both nucleic acids. They're both made out of nucleotides. Would you agree with that? Yes. DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose as the sugar, RNA has ribose. DNA has thymine instead of uracil. So if you see a T, it is only DNA. If you see a U, it is only RNA. DNA cannot have U, RNA cannot have T. That means rRNA, mRNA, tRNA, microRNA, none of them will have T's. They'll only have U's. That's it for that. How is transcription different in prokaryotes versus eukaryotes? List all the ways. There's the codon chart. All right, so I would say transcription, the goal, remember, is going from DNA to RNA. I would say initiation in prokaryotes is a lot more simple than eukaryotes. RNA polymerase just binds directly to the promoter, and then transcription starts. It's just so easy. But with eukaryotes, you have, remember, if you're pulling chapter 15 into this, you have activators first. Then transcription factors, they're purple bubbles, so they have to be proteins. Transcription factors bind to the ta-ta box of the promoter. The ta-ta box is part of the promoter. Then RNA polymerase binds. So that's a difference right there. Ta-ta. Elongation is the same for both. So elongation is where RNA polymerase is going to build RNA based on the DNA template. If DNA has a T, then mRNA has an A. If DNA has an A, then mRNA has a U. If DNA has a G, then mRNA has a C. If DNA has a C, then mRNA has a G. We've done that. Termination is the same. It just reaches a terminator sequence on DNA. You don't even know what that sequence is. Just know that something in the DNA sequence tells RNA polymerase, okay, you can stop now. All right, so far, what are the differences between prokaryotes and eukaryotes in terms of transcription? It's just the ta-ta box transcription factors. Eukaryotes require that. The second way that it's different is that eukaryotes have what's called pre-mRNA. It's the mRNA that's made, but it's not ready yet. In prokaryotes, mRNA is ready. So this is really important, something for you to write down. Remember. It was on your quiz, and it was the bonus, but now it's not going to be a bonus anymore. It's going to be an actual question. Notice in this picture that transcription and translation for prokaryotes is happening in the cytoplasm. Notice in this picture that transcription and translation are happening in two different places for a eukaryote. Why is that important? Well, this means in a prokaryote, transcription and translation can happen simultaneously. The cell can start making mRNA and it can start translating it before the mRNA is even done. It's an efficiency thing and it's kind of cool. But eukaryotes can't do that because that would really set us up, I think, for like mistakes to be made. It's like going too fast and not checking work. And prokaryotes can do that because their work is easy. They don't have very much to do. But in eukaryotes, there's too much to do. You got to have a very controlled step-by-step process. So I just want you to know that transcription and translation both occur in the cytoplasm of a bacteria, of a prokaryote, and therefore they can occur simultaneously. So in eukaryotes, you have the editing. There's 3 things that pre-mRNA that happens to pre-mRNA. You have a 5' cap, a poly A tail, and splicing. And we talked about splicing. If you have questions about splicing, let me know. A spliceosome is these crazy looking RNAs right here, these little like RNA bins. A spliceosome is a protein that has a bunch of RNA inside of it. An RNA will go and it will cut the intron out of the sequence of the mRNA and it will paste the exons together. So here, a spliceosome is actually acting as a ribozyme because RNA is cool. Alternative RNA splicing, this is why you have introns and exons, because what was an intron for one protein might be the exon for another. So one gene can be alternatively spliced to form actually different proteins each time. It just depends on which protein's needed. So that's why 20,000 genes can make 100,000 polypeptides because of our alternative RNA splicing. It's kind of cool. I feel like, I don't know, I kind of got lost here. So what are the 3 steps in transcription translation? Man, y'all can look that up. How is pre-mRNA processed? I just said it. What is the significance of alternative RNA splicing? I just said it. Why is a spliceosome called a ribozyme? I just said it. What is the job of tRNA? All right, now we're on to translation. The job of tRNA is to interpret mRNA. If you are confused about what tRNA does and what the process of translation looks like, there's this really cool game. You just Google Bioman bioprotein synthesis and you play this amazing game. It will be the best experience of your life. And it will help you understand transcription and translation so much better. But that's what you're doing in the game. You're building an amino acid by having an mRNA run through a ribosome and be read by tRNAs. And tRNAs are bringing in amino acids. Okay? The amino acids are going to bond together with peptide bonds. Wobble is not on there. There is a question about anticodon, just like the one on your quiz. Look at your quiz where it said DNA is AAA. What is tRNA? There's going to be another question like that. So if you don't understand that, you need to ask me. You say, what is the anti? Oh, I'll just tell you. If DNA is, okay, if DNA is GAG, mRNA is then going to be CUC. So tRNA is going to be GAG. You got to come talk to me if you don't understand that at this point. Aminoacyl tRNA synthetase is not going to be on there. Ribosomes, I mean, we know that from chapter 4. Ribosomes are made up of rRNA and proteins. That's what they're made of. The sites. A is where the tRNA comes in and brings the amino acid. P is where it moves over and forms a peptide bond between amino acids. And then E is where it drops the polypeptide and it leaves. Like I said, if you want a good visual, play the game. It says, if you picture translation in your mind, you see the animation. Oh, I even said it. If not, maybe play the game again. I mean, I just cannot condone that game enough. It's the best thing on the internet right now. How do free ribosomes become bound ribosomes? Skip that question. Skip that question. What would be the best way for a cell to mass produce proteins in large quantities? Polyribosomes, having multiple ribosomes translating the same mRNA at one time. Okay, and then proteins fold, we know that. We skipped signal peptide and zip code and stuff. I'm going to give you a DNA strand, and then I'm going to show you a new DNA strand and maybe a new RNA, new amino acids, and you're going to have to say what kind of mutation it is. So the different types of mutation are substitution, deletion, or insertion. Remember, substitution, one base is replaced with another. So you could end up with a new amino acid. You could end up with a premature stop codon with substitution. You could end up with a silent mutation. It could end up coding for the exact same amino acid at the end of it all anyway. So that's substitution. Frame shift would be, sorry, I'm answering a text. My bad. A frame shift would be as if you have, and that's my chair making noises. Sorry, it's a weird noise. A frame shift would be if you had a deletion or an insertion and it totally shifts the reading frame. So all the amino acids afterwards are going to be changed. So a frame shift is more detrimental than a substitution because a substitution only changes one amino acid. But a substitution could still cause a disease. It could. For example, sickle cell anemia is caused by a substitution. What makes it so bad is that originally right here where my cursor is, it is a hydrophilic amino acid. It has a hydrophilic R group. It's going to do hydrophilic things. It gets substituted for a hydrophobic amino acid. So that makes the protein folding vastly different, especially because of the location of that amino acid. That's why hemoglobin gets all weirdly shaped. Where's the picture of the weird shape? Right there. One amino acid. You exchange a hydrophobic for a hydrophilic, and it all of a sudden distorts the folding of hemoglobin so bad that instead of having round discs for red blood cells, it makes these like half moon shape, sickled shape, and they're sticky and they clog and they just cause a lot of problems. All right. Oh, what is a mutagen? That's not on your test, but just so you know, a mutagen is anything that can cause a DNA mutation. Man, am I gonna have to pause again? Okay, let me see. Is chapter 15 down here? Can y'all see like all my stuff down there? You probably can. Ooh, let me pause. Okay, I couldn't find the PowerPoint on this device. It's on my other computer. So I just went to Veracross and got the PDF. If you ever see like a weird chapter number, so this is chapter 18. This is actually chapter 14 for your book. It was chapter 18 for the old book. So chapter 15. Chapter 15, yeah, for your book. Gene regulation is necessary. We said it early. We said it in class to, oh, that's not what I was trying to do. To prevent waste, man. Ugh. So the purpose of gene regulation is to prevent waste and to increase efficiency. So you don't want genes turned on that you don't need, and you don't want genes turned off that you do need. And so that's why it's necessary. Explain negative feedback as it applies to regulating metabolic pathways. Let me come back to that one. Prokaryotes are going to regulate gene expression using these things called operons. And remember, we have two types of operons, a repressible and an inducible operon. It's prokaryotes, not eukaryotes. So you don't do this. Two types, repressible and inducible. A repressible operon is on until turned off. The gene is on until it's turned off. The gene is transcribing and making something until it no longer needs that thing anymore, and then it gets turned off. An inducible operon is normally turned off until something is presented that turns it on. It's off because it's a gene that codes for something that's not always needed. So the example, okay, so those are the two. A repressible operon is always on because it's always, there's something that it codes for that's always needed, like amino acids. Like it's coding for one amino acid. So you always need it to be on until it makes too much of that thing. Then you turn it off. It's kind of like, oh God, I'm about to say an analogy and I hope it's not bad. You drink water every day. You always drink water, right? Every single day you're gonna need water. But you can't drink too much water. At some point you're gonna like put the water bottle down, right? But every day you're always gonna need to drink water. That's kind of like what a repressible operon would be, like what the genes it codes for. It's something that's always needed no matter what. So it's normally turned on. You're normally gonna have access to water all the time. There's water fountains everywhere. I hope that doesn't confuse people more. So it's on until turned off. The example is the tryptophan operon. Tryptophan's an amino acid. Once enough tryptophan is made, the tryptophan itself acts as the co-repressor, binds to the repressor, that little Lego piece that then fits into the operator and blocks RNA polymerase from transcribing anymore. It's like, we've got enough tryptophan now, chill, and so it stops. Always look at the slideshows if you need like more information and more pictures. I would always supplement looking at your notes with looking at the slideshow. An inducible operon is the opposite. It's normally off until turned back on. An inducible operon is when there's a gene that's really not always needed. So it's kind of like eating chocolate cake. At some point, you're going to need to eat some chocolate cake, but you're not doing it every day, you know? So the bacteria, sometimes they're going to need to make lactase, but they're not going to do it all the time because the sugar, lactose, isn't always present. So it's more convenient to just have that gene always turned off until something comes in, the sugar, to turn it on. It's like, hey, we're eating lactose today. Yes. Let's make some lactase so we can digest it. Because remember, lactose is a disaccharide. You got to have an enzyme to break the bond so that the actual monosaccharides can be used for energy. So this little green pea looking thing, that is the inducer. So in this example, lactose, the sugar, is present in the environment. So lactose comes in, pulls the repressor off, and basically gives permission for the gene to be expressed and for lactase to be made. That's an inducible operon. Okay, that's how prokaryotes regulate genes, blah, 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 blah. An example, hey man, talk through the picture, all right. We're gonna move on to, no, click. Oh, it doesn't click, I have to scroll, sorry. Differential gene expression just means depending on what type of cell it is, it's only gonna express certain genes. And the genes it's gonna express are the ones that are gonna help it do its job. Gene expression in eukaryotes is controlled at 4 different stages. The first stage is chromatin structure. Methylation, it makes DNA more hydrophobic, so then RNA polymerase cannot access it, so transcription is decreased. The genes are not expressed. Histone acetylation, remember acetyl groups are hydrophilic. It relaxes and unpacks DNA, making it more accessible to RNA polymerase. So transcription is increased. That's at the chromatin level. Epigenetic inheritance is methylation and acetylation being passed on to subsequent generations. And we talked about the mice and the fruit. It's kind of interesting. I don't think I didn't ask you any. I mean, I would know just the definition of epigenetics. So modifications of chromatin that can be passed down to future generations. So methylation and acetylation can be passed down to generations. There's some cool videos. If we had our lab period, we could have talked about that a little bit more. God. Ugh. Okay, I'm not. We could have talked about that more, but I have to cut so much stuff. It is what it is. All right, next. Second way that gene regulation occurs in eukaryotes. I'm on the last page of the review. So it's telling you control elements and enhancer region. Just cross those questions out. I'm not going to get that nitpicky. My tests actually used to be that nitpicky. I've gotten a lot nicer, believe it or not. But my test, it's just the question bank changed. So it's actually the questions that were mean, not me. I just took whatever questions I could get because I'm not going to write my own. So because those were the ones I was told to use by College Board. But they changed their questions, so then I changed mine to be a little bit more broad in scope and not quite so detailed. So control element and enhancer you don't need to know. What do I really just want you to know is that you have this thing called a transcription initiation complex in eukaryotes that requires activators to be there so that transcription factors can bind to the ta-ta box, mediator proteins can stabilize the structure, and RNA polymerase can bind. And so there's a question that's going to ask you something about this. Well, what's happening in this picture? Well, you have a liver cell on the left side and a lens cell on the right side. You have the liver cell expressing the gene for albumin, but not crystalline. And you have the lens cell expressing the gene for crystalline and not albumin. Why is it that the liver cell can make albumin and the lens cell can't? It's because, and look at the color coding, red, gray, yellow. Does the lens cell have the red, the gray, and the yellow? It has the gray. Cool, that's not enough. It's trying. You see it trying there? The gray one's like, I'm here. Where's everybody else? They're not there. The yellow ones and the red ones aren't there. So it cannot even initiate transcription at all. Albumin's not made in the eye. But the liver cell has the red, the gray, and the yellow. It can initiate it, so therefore transcription for albumin, the protein, is going to happen. What about on the flip side? Crystalline requires a pink, a gray, and an orange. The lens cell has pink, gray, and orange activators. And these activators are just little teeny little proteins that bind to this part of DNA, helping transcription get started. So what's the conclusion? Depending on the activators present in a eukaryote cell, that will determine the genes that are expressed. The activators have to match that certain gene. Hope that made sense. Okay. That's the last page on the review, but I do have 2 more things to say. Third level of gene regulation in a eukaryote has to do with microRNAs degrading and chopping up mRNA that's no longer needed. So microRNA is going to chop up mRNA that's no longer needed. In order to chop something up, it means it has to break bonds. If something can break bonds while staying unchanged itself in the process, isn't it acting like an enzyme? It sure is. So this is another example, in addition to spliceosomes, of how RNA can act as an enzyme and how RNA is really awesome. And it was probably the first genetic material. I know I said that, but I always like to bring that up because I see that question being asked at the end on the AP exam. Fourth way that gene regulation occurs is when the protein's already made. I don't know if I would even call this gene regulation, but it's the product of the gene. And if you're chopping up and degrading the product of the gene, I guess in a way it is. So the protein was made, the protein was used, it's no longer needed. So let's say you eat that chocolate cake that I was talking about a little while ago. And so your cells are signaled to make insulin. Insulin is going to control blood sugar, and it's going to bring sugar into the cell so it can be used for energy. Let's say the next day you feel so guilty about eating that chocolate cake. You're like, I'm going on the keto diet. I'm doing it this time. And you stop eating sugar completely. Your body's going to be like, well, I guess she's on the keto diet again. We don't really need this insulin for now. Let's go ahead because we don't want insulin unused just floating around. That's useless. It's taking up space. It's blocking traffic. So proteasomes are going to come and they're going to chop up the protein that's no longer needed. In my example, that protein was insulin, until I start eating chocolate cake again, and then it's going to make more insulin. That's it. I gave all the hints. Hold on, let me look at my little paper here. Ooh, girl, I gave all the hints. Yep, I did. All right, I'm done. Thank you for watching.